I'm Dr. Ashwini Kumar, Director of Vector Control Research Center at Puducherry. And the topic that is allotted to me is the dynamics and modes of transmission of vector-borne diseases. Okay, I believe that by now you have been exposed to the modes of transmission of infections. There are two kinds of infections. They are categorized into direct transmission and indirect transmission. In the direct transmission category, there are 5 subcategories known. Number 1, direct contact. Number 2, droplet infection. Number 3, contact with soil. Number 4, inoculation into skin or mucosa. And number 5 is transplacental, which means vertical transmission from mother to the child. This is one of the classifications of infections, and the indirect transmission includes vehicle-borne, example of which is the birds taking infectious agents from one continent to another, one country to another, or import of animals that can also bring in infectious agents, or vectors which are attached to them. And number 2 is the vector-borne transmission, which is also an indirect transmission of infectious agents. Number 3 is airborne, which is droplet nuclei, which are dried nuclei present in the soil. And of course, fomites. Fomites means any objects, maybe table, chair, glass, anything on which the infectious agent is attached. And 5 is unclean hands and fingers. Hand transmission of infectious agents is also very well known. I hope this is clear. So we will focus on vector-borne disease. And well, out of total infectious diseases, the vector-borne diseases constitute about 17% as per the World Health Organization. And the prominent vector-borne diseases in India are malaria, filariasis, leishmaniasis, dengue, chikungunya. Of course, Zika is now emerging, Japanese encephalitis, Kyasanur forest disease, popularly known as KFD, and scrub typhus and tick typhus, which are not so well known, but are present and diagnosed from time to time. And scrub typhus is definitely fast emerging infection in our country. This is a central triad, which is a famous triad of host, vector, and parasite or pathogen. The pathogens are moved from host to the next host via vectors. And the environment regulates this transmission. The environmental conditions are very, very important in not only maintaining the cycle of transmission, but also the seasonal outbreaks of transmission. Let us see how we can classify the vector-borne diseases. There are various epidemiological classifications. Number 1 is by vectors. Now, there are invertebrate vectors and there are vertebrate vectors. The invertebrate vectors, which will be the focus of my talk, they are dipteran. Diptera order includes flies and mosquitoes. Siphonoptera includes fleas. As you know, fleas cause plague. Mosquitoes cause malaria, filariasis, dengue, chikungunya, Japanese encephalitis. And number 3 is Orthoptera. These are cockroaches. Cockroaches transmit various gastrointestinal infections and allergic causing germs, and they are in Orthoptera. Anopleura, sucking lice, head lice, etc. Hemiptera, which has bugs, including the kissing bugs, causing diseases in the South American continent. Acarina, which includes ticks and mites, and ticks, as we know, cause, for example, KFD and also tick typhus. Mites cause scrub typhus, and copypods, cyclops, are also invertebrate disease transmitting vectors, and they are implicated in cholera. Vertebrate hosts include mice, rodents, and bats. And mice are implicated in transmission of several infections. Rodents likewise are also the vectors of several viral infections. Bats are known to harbor various coronaviruses. So these are also examples of vectors, but they are vertebrate vectors. By transmission chain, we can divide into 3 different kinds of transmission dynamics that are known: man and an invertebrate host. That means number 1, man, arthropod, man. This infection is, we all know, is the example of this is malaria, filariasis, and dengue. From man to man via arthropod vector. Man, snail, man, schistosomiasis. And in B, there is another vertebrate host involved. For example, a mammal, arthropod, and man. And this is an example of plague. And the second is bird, because in the plague, rats are involved, and of course, fleas transmit Yersinia pestis from mammals to humans. Then birds, arthropod, and man. For example, Japanese encephalitis. Birds are amplifying hosts, and they amplify Japanese encephalitis viruses. We will come to that later. And the C, man and 2 vertebrate hosts, the examples of which are man, cyclops, fish, man, fish tapeworms, then man, snail, fish, man, Clunorchis sinensis, and man, snail, crab, man, Paragoniomyces. This is an example of 2 intermediate hosts involved in the transmission. Now, there is another classification by the method in which the vectors transmit agents. So, number 1, by biting. Number 2, by regurgitating. Number 3, by scratching in of infective feces. And number 4, contamination of host with body fluid of vectors. So this is another classification of the transmission, modes of transmission: by biting, by regurgitating, by scratching in of infective feces, and contamination of host with body fluids of vectors. Now, yet another classification is by the method of transmission and propagation of parasites or pathogens inside the hosts. In this, there are two categories. One is the mechanical transmission. That is, the pathogens are attached to the legs or the other body parts of the vectors. They are not internalized. They are simply transported from the infectious surface or feces to the food or water. And examples of this are gastrointestinal infections, typhoid, cholera, through the flies and also through crawling insects like cockroaches. These are mechanical vectors. And the biological vectors in which most of our vector-borne diseases fall that are of concern to us, we have three categories. One is propagative vectors. In these, for example, dengue and plague, in this, the disease agent multiplies but does not change its form. It simply multiplies. Number two, cyclopropagative, in which agent not only multiplies but also changes its form. For example, plasmodium. Plasmodium changes several forms during its life cycle in the vector itself as well as in the human host. Cyclodevelopmental. In this case, for example, in lymphatic filariasis, in mosquitoes, larvae simply develop from microfilary to L3 stage, which is an infective stage, but without multiplying. So we have learned that in this particular case, there are two kinds of vectors: mechanical vectors that they cause mechanical transmission by simply transporting the pathogen from the infected surface to the food or to the water and cause infection by simply ingestion. And in case of biological transmission, either it is propagative. For example, in case of plague, as I said, and dengue, there is a multiplication in chikungunya, in Zika, in KFD, same case, there is a multiplication without changing the form. In cyclopropagative, the agent multiplies and also changes its forms, like leishmania, like plasmodium. In third category, the larva simply develops, as in case of lymphatic filariasis, the LF larva only transforms itself from microfilary to L3 stage, which is infective. So the natural history of vector-borne disease starts with the exposure to the infectious bite or contamination of food or water, that is mechanical transmission, as I mentioned. And you need a susceptible host to whom these infectious agents will infect and cause vector-borne disease. And there will be three outcomes. One is the recovery. The second is short or long disability. For example, in case of lymphatic filariasis, there can be chronic LF disease and may cause a long-term disability, or a short disability in case of malaria, 3 to 4 days, and then recovery, or 7 days, then recovery. Or there can be severity, and severity can lead to death. So these are the three outcomes and the natural history of infectious diseases. Let us now look at the examples of the major vector-borne diseases. Malaria is the king of all the diseases. It was called once upon a time king of all diseases, as it would kill several hundreds and thousands of people in the world more than all the wars that have been fought on this earth. But now in India, we are trying to eliminate it by the year 2027 and certification by 2030. So this is a life cycle of Plasmodium parasite. There are 4 human parasites: Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae. Plasmodium falciparum causes complicated malaria. Plasmodium vivax and ovale, they have relapses. Plasmodium malariae tends to cause a chronic infection. And Plasmodium falciparum and vivax, these two are by far the common species. And in our country, there are several vectors involved in the transmission of Plasmodium. For example, Anopheles culicifacis in the rural areas, Anopheles fluviatilis in the foothill areas, Anopheles minimus again in the Tarai belt and in northeast, and also it has been found in Odisha also in the foothill areas. Stephen's eye, which is a urban vector, and Baimai, which is primarily in the northeastern states, and Sandaicus, Anopheles sandaicus, which used to be in Odisha around Chilka Lake. Now it is no more available in that earlier area of its distribution, but is now restricted to Andaman and Car Nicobar Islands. Now here you would see that only one host, the human host, is involved as far as these 4 parasites are concerned, and the vector picks up infection from the human when they bite, and gametocyte stage is internalized, and of course then there is a development within the vector host. So this is cyclopropagative. Not only there is a development, there is a change in form also. This is an example. And in human being also, sporozoites develop in liver, and then finally when they come out in the form of merozoites, they start a retrocytic cycle, which is repetitive cycle, and finally some of them will make gametocytes, which are picked up by the vector, and the transmission will go on. So this is cyclopropagative transmission. True vector is involved, only one host, and of all these 4 species. There is also 5th species now, Plasmodium knowlesi. Plasmodium knowlesi is found in large numbers in humans in Malaysia, and occasionally they have been reported in India, in Andaman and Car Nicobar Island, and also in the northeastern state. So a lot of research is needed to really know the distribution of this species. Likewise, Plasmodium ovale is also a rare species in India. Plasmodium malariae is found in pockets, sometimes in association with falciparum and vivax, and it is very much found in certain districts of Odisha. For example, in Sundargarh, where I worked in 1989, we used to get good number of Plasmodium malariae in association with falciparum mixed infections with vivax falciparum also. And this is, of course, a neglected malaria parasite species. Let's go to the next one. This is the life cycle of Leishmania. As you know, India is also eliminating Leishmania. And Leishmania has very restricted distribution. Leishmania donovani, we call it Kala Azar. This Kala Azar disease has presence in certain districts of Bihar, northern Jharkhand, eastern UP, and some districts of West Bengal. The vector is Phlebotomus argentipus, and in certain parts, Papatasi is also suspected. And in New World, it's Lutzomia species that transmit Leishmania in Donovani infectum and Brasiliensis. So in India, we find only Leishmania donovani, which causes VL infection, and post-Kala-Azar dermal leishmaniasis. The sandflies picks up the A. mastigotes and ingests the macrophages, and then of course these parasites are released, and they are upon ingestion and transform themselves into promastigotes, which multiply and then lodge themselves in salivary glands of the fly. And finally, when this fly bites the new susceptible host, next it injects the Leishmania into the next host, and then of course Leishmania changes its form and finally lodges itself in macrophages, and the cycle continues. And here it is to be noted that new foci of Leishmania donovani have emerged in Uttarakhand, in Himachal Pradesh, and our field station in Kottayam has found several pockets of VL and CL transmitted by Leishmania donovani MON 37 strain. Interestingly, this species in Kerala causes both cutaneous leishmaniasis as well as visceral leishmaniasis. So here also, it's the direct transmission from human via the vector to the next human host. It is now suspected that some mammals may be involved as reservoir, like dogs and shews. And in Kerala, for example, our team has found Leishmania donovani in dogs. So it appears that the reservoir host maintains Leishmania donovani, certain strains, and help in transmission of the parasite. As far as filariasis is concerned, again, this is also a direct transmission, but cyclodevelopmental, example of cyclodevelopmental propagation and transmission. Here, the parasites, which are adults, male and female, are lodged in the lymphatics of the human host, and they produce large number of microfilariae. These microfilariae then come into peripheral circulation and picked up by the mosquitoes, and primary vector in this case is Culex quinquefasciatus. As far as Wuchereria bancrofti is concerned, as far as Brugia malai is concerned, it's Mansonia annulifera and Mansonia indiana and uniformis, which are associated with the seaweeds or water hyacinths in the brackish waters. In Odisha, you have a focus in Baleshwar of Brugia malai, in addition to Wuchereria bancrofti, which is quite prevalent or endemic along the coastal districts of Odisha. So the vector picks up the microfilariae, and these microfilariae would immediately shed sheath, and they will convert into L1 larva, and then L2 and L3. L3 then migrates into the head region and finally into proboscis, and when this infective vector bites the host, the parasite is injected into the injured part, and from there it moves into the peripheral circulation, and finally this L3 larva converts into adults in infective form, and then goes in converts into adults, and then they lodge into lymphatics, as I mentioned you earlier. This is how the cycle goes on. This is the life cycle of dengue. Now, dengue is caused by flavivirus, and there are 4 serotypes known: DEN1, DEN2, DEN3, and DEN4. And the transmitting species involved are Aedes aegypti, which is urban vector, and sylvatic vector albopictus. Both are known as efficient vectors of dengue. Aedes aegypti by far superior vector than Aedes albopictus, and Aedes vittatus also has been incriminated as the third species which is playing some role in the transmission of dengue infection. What happens in this case is that when a mosquito bites any infected person, the mosquito which carries the infection here is, as we know, is a diurnal biter. It bites during the day, and it is a nervous feeder. It is called a nervous feeder because it has to survive, it has to escape the attention of host, and it has to bite. So it will take a part of its blood meal when it bites, and then flies, and then goes on biting several hosts before it gets the full blood meal, and in the process can infect several hosts, human hosts. So you will find often dengue will be a cluster disease where it can infect many. And also, interestingly, there is a vertical transmission from mother to the next progeny, the daughters. That is called trans-ovarian transmission. It is well known in case of dengue viruses. And different serotypes may be distributed in different geographical areas, and in a single geographical area you may see different serotypes surfacing in different years, and sometimes 2 or 3 serotypes may be transmitted in the same area. So when these viruses multiply, then obviously they from the gut they lodge themselves into salivary glands, and it's a very simple cycle that transmission occurs. In case of chikungunya virus, which is an alpha virus, I hope you remember in 2006 and 2007, there was a huge outbreak of chikungunya virus in the country, and several districts of Kerala were very badly affected due to chikungunya. Chikungunya is actually an African virus which was first reported in 1957 in Uganda. And chikungunya means that bent posture. And it causes a lot of myalgia and pain in the joints. And this virus is transmitted by, again, same vectors, Aedes aegypti and Aedes albopictus. And in Africa, it is known to circulate among the non-human primates like chimpanzees, monkeys, and baboons, and maintain a sylvatic cycle. Now, there the species involved are Aedes africanus, Aedes falciparum, and another Aedes species is involved in the maintenance of sylvatic cycle. However, in India, we are not yet aware, we do not know whether there is a sylvatic cycle of chikungunya present. The next one is Zika virus. Now, Zika virus surfaced in 2007 in the Latin America, and in Brazil 2015 there was a big outbreak of Zika virus, and here the cycle is a bit complex. Although the vectors are Aedes aegypti, and the primary vector is Aedes aegypti, in Brazil, Aedes albopictus and vittatus have been implicated in our country. I'll tell you shortly. Now, let's just look at the cycle. The virus can be transmitted by vector from human to human, that is a human cycle, and from male to female or female to male by sexual transmission. Sexual transmission is also known, and from mother to child vertical transmission is also known. And the children that are born to the infected mother in Brazil had microcephaly. That means the brain was small. And they also called what is called the another symptom, there is Guillain-Barré syndrome is there, which is also seen in the infected persons who get infected. And there is an enzootic cycle wherein the mosquito can maintain this cycle in rhesus monkeys, and in monkeys also sexual transmission is also suspected. And also there is a suspicion that there are reservoir hosts in the domestic animals of Zika virus. Now, the recent story about Zika virus transmission in India, you might have read it in the papers, was in Kerala state, in Thiruvananthapuram, where several cases were reported from a hospital, tertiary hospital, where one case was admitted and then subsequently several cases were detected. And our team collected several vectors, Aedes aegypti, Aedes albopictus and vittatus, and isolated virus from these vectors as well, which led to the confirmation of not only its transmission, but also involvement of 3 species as against 1 species which was suspected to be involved initially, Aedes aegypti. As far as Japanese encephalitis is concerned, this causes sporadic infections, and we know that vaccine is available for pigs as well as humans. In case of Japanese encephalitis virus, it's a flavivirus, and Culex vishnui group of mosquitoes transmit the virus. Virus is maintained in nature between vector and the pigs. This is the natural cycle. And also, and pigs are amplifying hosts. And similarly, paddy herons and cattle egrets, they also amplify virus. And there can be transmission between these 2 cycles. The vector can bite alternately pigs or birds or vice versa, and this cycle is maintained. But during monsoons, when large areas are under paddy cultivation, there is expansion of this vishnui group of mosquitoes, and we see that the infection in humans is noted. and humans are dead end. Humans and cattle or horses, other animals are dead end. They cannot further transmit the disease. Let us talk about Kyasanur forest disease. The Kyasanur forest disease, or KFD, was originally detected in Shimoga district of Karnataka, and subsequently, and that was associated with the monkey deaths, and subsequently the infection spread not only to the neighboring areas of Shimoga, but also till Wayanad in Kerala and Maharashtra in 2015 and 16, Maharashtra and Goa, where the KFD related deaths were reported. Here, the ticks produce large number of eggs during monsoons, and they produce the larvae and the nymph during November, December in the dry season. They resort to biting various animals, shrews and monkeys and all that. And then when the infection occurs in monkeys can die due to KFD, and the nymphs that are on these monkeys, ticks would leave the monkey and can cause infection in humans that will come in close contact with the monkey, or the visit to the forest for forest produce or for cattle grazing, and they would also get infected, and some of whom would die. The cattle helps in maintenance of the tick population, the adult ticks population, and these ticks would produce eggs, as I mentioned, in the foliage, and this cycle would continue. And the primary vector species is Haemaphysalis spinigera, and Haemaphysalis tertius is the secondary vector known. It's a flavivirus, and it is an expanding and emerging disease. So to wrap up today's lecture, what we learned is that about 17% of infectious diseases are vector-borne, with 700,000 deaths annually in the world. Of course, many vector-borne disease-related deaths are also reported in our country. India is currently eliminating malaria, lymphatic filariasis, and visceral leishmaniasis. However, dengue is the fastest emerging arboviral disease, not only in India, but also rest of the world, several countries. Vector-borne diseases are caused as a result of indirect transmission involving vectors, disease agent, reservoir, and or susceptible human host. In India, the prevalent vector-borne diseases include malaria, filariasis, leishmaniasis, dengue, chikungunya, Zika, Japanese encephalitis, and KFD, besides lesser known rickettsial diseases like scrubs and tick typhus. The biological mode of transmission includes propagative, cyclopropagative, and cyclodevelopmental modes of transmission. Thank you very much for patient hearing.